hope I'm like audible and visible, right?
Yes, sir. Kindly share your screen, sir.
Okay, perfect. So I hope I'm audible, visible. Everything is like fine. Yes, right? Shall I start?
Yes, sir.
Okay, perfect. So yes, good evening, everyone. This side Dr. Abhijeet, and welcome to yet another session of this 45 days to MBBS final exam. And this intensive revision series, I will be focusing today on the mock paper discussion that we already had with you guys, right? So yes, I will be revising all the important questions that are like being asked. Obviously, in the mock paper, we have given you the most expected questions or the most potent questions, right? So I will be discussing that right now. Apart from that, guys, you already know why to choose Oracle, right? We have the recorded classes along with the live classes. The practical sessions are also there. The exam modules are also there. And even now we have the hard copy book set that you can like order from your home. And yes, it will be in home delivery, right? Apart from that, guys, all these recorded classes, live classes, express exam modules are there. One-to-one doubt clearing sessions are there, right? The practical sessions are being recorded over here. Apart from that, we have a very customized question bank that basically offers no all the questions basically they're asked from basic to advanced level. And you guys already know that yes, the questions basically cover the last 10 years, right? The 10 years, yes. The important questions that have been asked in the previous 10 years in the NEET PG exam or like any national exam of the country. Apart from that, we have these hard copy books that I have already told you. Now, this is the basic subscription plan, guys. You can just order this like it's the basic plan, standard and premium plan. And apart from that, guys, if you want some extra discounts, you can just use the code Dr. Abhijeet 20 for further discounts, right? Now, let us start with the biochemistry test paper that is a total of marks 100. And yes, this paper is already with you guys. And let us just try to start with the very first question. And guys, remember that this is the question that has been asked in like asked multiple numbers of time, right, in various university exams. And this is something like which I have myself discussed in this the last week, right? So the question says that describe the beta oxidation pathway of fatty acids, right? Earlier, the question that I discussed, it was like, yeah, the palmitic acid, right? But they have simply asked you any beta oxidation of the fatty acid. I have to tell you about its regulation and the significance of ketone bodies formation during prolonged fasting. So they are asking like three parts of this question, this 10 marks question. They have like three parts, guys. Firstly, I have to tell about the all the like steps of the beta oxidation. Apart from that, we have to discuss the regulation, guys. And apart from that, we have to discuss the significance of ketone bodies formation, right? The prolonged fasting, basically, they are asking about starvation ketosis. They are asking about starvation ketosis, right? Now let us just start with this. Now, guys, this is something that we have already discussed. Now, oxidation of any saturated fatty acid. Okay, saturated fatty acid. For example, if like we are taking palmitic acid, or it can be any fatty acid, guys, right? Firstly, you should know beta what is the site. I have already told you how to write a question in the exam. That is something very, very important. They're not asking you to write thesis beta. The answer should be like pinpoint, like it should be concise. I mean, all the informative things should be there. You don't have to write stories, no, to make it lengthy or something. Apart, unless and until it like carries all the important information, that questions makes no sense, right? So all the important information should be concise and it should be like accumulated in like yes. The sites are the liver, guys, the adipose tissue, and the muscle. Organelle you are going to write, it is the mitochondria. And since they have asked the beta oxidation pathway, so you are going to describe all the steps of the beta oxidation, all the steps of the beta oxidation. The very first step, it says that activation of fatty acid, right? Activation of fatty acid. Then we have the transport of activated fatty acid to mitochondria. And then we have the proper beta oxidation. This is, guys, something that we have already discussed in the last week. Now, activation is something very, now any fatty acid, guys, it can be palmitic acid or any fatty acid, when, okay, it will be getting converted to acyl coenzyme. That is called as activation of fatty acid. And remember, guys, you have to mention this for activation, guys. Activation starts with A, and over here, ATP is required. That is an energy utilizing step. Okay, and two high phosphate energy bonds are required for to activate this fatty acid, right? And that is by the enzyme acyl coenzyme synthase, that is called as thiokinase. That's a part of thiokinase, right? Now, guys, this particular step requires two high energy for phosphate. You have to write all this in your exam, guys. These are all important markers that you have to like mention in your exam. You have to write about that this enzyme, the acyl coenzyme A belongs to ligase, right? And ligase, you already know it needs ATP. It needs ATP. Okay, it is the only energy requiring step in fatty acid oxidation. I've told you this is the first step and the only step that is requiring energy. The two high phosphate molecules. You can see that ATP, adenosine triphosphate, it is converting into adenosine monophosphate, right? So tri is converting into mono. It clearly means that two phosphates are required. Now, this particular step is something which is like taking place in the cytoplasm, right? Okay. And this enzyme, acyl synthase, where is the location, guys? You also have to mention it is located in the outer mitochondrial membrane. It is located in the outer mitochondrial membrane. Now, guys, transport of activated fatty acid. I have already told you about the carnitine transportation, right? The carnitine transportation from outer mitochondrial membrane to the inner mitochondrial membrane and ultimately to the, yes, to the matrix, right? So that is the, yeah, the carnitine which is used as a transporter over here. Apart from that, the third one, that is the beta oxidation proper, that is like taking place in the mitochondria, guys. There are four steps. You already know acyl coenzyme A. Then we have enoyl coenzyme A. FADH2. Reducing equivalents are released over here, right? Dehydrogenase. Then enoyl coenzyme A converts into hydroxyl acyl coenzyme A. Obviously, water is added over here. And then hydroxyl acyl coenzyme A, it is converted into keto acyl coenzyme A with the release of another NADH, another reducing equivalent that gives ATP. And then apparently, we have acyl coenzyme A, which is like less than 2 carbon. That 2 carbon, it is in the form of acetyl coenzyme A. This 2 carbon will be broken down, right? For example, we have like palmitic acid. We have 16 carbons. 2 carbon will be removed. This cycle. Again, this cycle will be repeated. Again, this cycle will be repeated. Now, this 2 carbon, which is removed in the form of this acetyl coenzyme A, it will go into the TCA cycle to generate energy, right? To generate the ATP, right? So ultimately, TCA cycle is the common oxidative pathway for all the macromolecules, whether it is carbohydrate, proteins, or lipids, right? So you have to write better all these three important steps: activation, transportation, and beta oxidation proper. In this particular first question that is asked, describe the beta oxidation pathway of fatty acids. Very, very, very important, guys. Very, very, very important. Now, coming in onto its regulation, guys. Coming onto its regulation. What is like the regulation part, guys? You have to remember. Okay, what all points you have to like mention in the regulation, guys? Regulation. Now, guys, remember that regulation basically, yes, you already know when there is a well-fed state. Now, when there is a well-fed state, you already know well-fed state, it is just 0 to 4 hours after food. 0 to 4 hours after food, right? It is the well-fed state, that is 0 to 4 hours after food. Now, in well-fed state, you already know which is the hormone, that is the insulin is the hormone. Insulin is the hormone. You already know this insulin. Yes, insulin is the hormone. And guys, remember this, there will be, okay, there will be increased fatty acid synthesis over here. There will be increased fatty acid synthesis over here, right? There will be increased fatty acid synthesis over here. You already know, yes, if insulin is the hormone, there is always high insulin glucagon ratio. high insulin glucagon ratio, and that will something which will increase the fatty acid, right? And remember, guys, over here we have malonyl coenzyme A, which is like an elastic inhibitor of CPT1, right? So the CPT1 is like closed, right? Now, apart from that, yes, another regulation, guys. One is in the well-fed state. Again, in fasting state, what happens, guys? In fasting state, you should remember that glucagon is the hormone. Glucagon is hormone, right? Now over here, insulin is the hormone. Over here in fasting state, glucagon is the hormone. We have low insulin glucagon ratio. We have low insulin glucagon ratio. And remember, guys, there is decreased level of malonyl coenzyme A. Malonyl coenzyme A. And so what will happen? That CPT1 will activate, right? Or will open. So that will like, yes, it will be decreased, right? So that is how you have to explain the regulation, guys, over here. That is how you have to like focus on the regulation part over here, right? So that is like two important points in the well-fed state, how it is regulated, and in the fasting state, how it is regulated. Like in simply, you have to mention these important points in that question. Now they have asked the significance of the ketone bodies. Yes, now they have asked the significance of the ketone bodies during prolonged fasting. Now, guys, you already know that is something called as starvation ketosis. Now, in prolonged fasting, guys, let us take an example. You already know when it is like 2 to 5 days, 2 to 5 days, and you are not taking any food. You are not taking any food. There is no food, guys. That will something lead to starvation, or it is also called as prolonged fasting. Starvation or prolonged fasting, right? Now, what is happening? What is happening, guys? You already know. Initially, when there is a well-fed state, you already know the insulin is the hormone. Okay, the okay glycogenesis will take place. Obviously, the extra glucose which is like there, it will be converted into glycogen. In early fasting, in early fasting, this we have glycogenolysis. Obviously, it is like 4 to 16 hours after food. All the glycogen that is being stored in the body, it will be broken down to provide energy, right? After 4 hours, you again not start eating again, right? Now, after early fasting comes late fasting. Late fasting, that is like more than 16 hours, right? Till 2 days. And over here, when you are not eating any food, guys, the body will try to make glucose from some non-carbohydrate substrates. And that is gluconeogenesis. Gluconeogenesis will be like, it will be using oxaloacetate. Okay. It can use other substances as well, right? Oxaloacetate will be used to form glucose. Now, guys, here's the clinch. You already know oxaloacetate. It's a part of TCA cycle. It's taking part in the TCA cycle. Now, in late fasting, like more than 16 hours, you're not taking any food. The oxaloacetate is also depleted. Now, glycogen depleted, gluconeogenesis substrate, that is oxaloacetate is also depleted, right? Oxaloacetate is also depleted. Now there will be starvation, right? Now what will happen? There will be beta oxidation of the keto acids. You already know triacylglycerol will be broken down into fatty acid. Fatty acid will be broken down into acetyl coenzyme. Now this acetyl coenzyme, obviously fatty acids are, you already know, we have a 2 carbon less from 16, then again repeat cycle 2 carbon less. This acetyl coenzyme is a 2 carbon compound. It will try to enter the Krebs cycle, but it unfortunately cannot enter because in TCA cycle, oxaloacetate is not there. It's already vanished. It will now go on to ketone body synthesis, and that is how they have like asked this a very simple question that you should remember, guys, over here. That is the significance of the ketone bodies during prolonged fasting. So in that case, guys, this is how the regulation takes place. This is how all the steps are there. And guys, you have to like, yes, mention about the starvation ketosis as well. So that is a complete 10-mark question, right? That is how you have to write, mention in your exam, all the flow charts, all the enzymes, right? All the reducing equivalents, all the energy equivalents. I have to just mention like that. Now, coming on to the next part, guys, the next question. Explain the biosynthesis of purine nucleotides. Discuss the role of key enzymes involved in the regulation of this pathway. Now, guys, you already know about this, that, yes, yeah, guys, you already know about this, that we have already like discussed previously also. Yeah, about the purine metabolism, right? How this purine nucleotide is synthesized. Firstly, you have to see where all these structures are coming from. Now, you already know the structure of purine, right? You already know the structure of purine, right? We have nitrogen at all the odd places except the fifth one, right? Except the fifth one, starting from here, 1, 2, 3, 4, 5, 6, right? And yes, you already know we have nitrogen at seventh place also. Yes, and also at ninth place. Ninth place, right? Seventh, right? Eight and nine. Now this is the basic structure that you have to know. Now again, you can draw the structure and write the precursors. That is again like you can draw this like very easy. I've already told you, right? First you have to go in the clockwise direction, then in the anti-clockwise direction, right? No, first in the anti-clockwise direction and then in the clockwise direction. Again, the numbering is very easy. Now, guys, this C, it is basically coming from the respiratory CO2, right? This nitrogen, at the first plate, it is coming from the aspartate. It is coming from aspartate. It is coming from aspartate. It is coming from aspartate. Yes, it is coming from the aspartate, right? Now, this carbon, it is coming from N5 formyl tetrahydrofolate, right? Now, these two nitrogen at third place and the ninth place, it is the amide nitrogen of glutamine. Amide nitrogen of glutamine, right? And this is the eighth one, the carbon, it is coming from N5,9,10 methynyl tetrahydrofolic acid. And yes, these two carbons and nitrogen, it is coming from glycine. It is coming from glycine. So this is the precursors that are now basically used over here, right? Then we have the ribonucleotide formation. You guys already know the purine bases are synthesized as ribonucleotides, right? So this is like you have to mention the ribo. Yes. Now, guys, firstly, it is the PRPP formation. You guys already know how you have to write down this, guys. Please try to write down like this in the form of a flow chart. Again, you can like mention as this also, which is written in the notes. But otherwise, you can just mention like this also, ribose 5-phosphate. It will be converted into, yes, what is PRPP? It is phosphoribosyl pyrophosphate. Pyrophosphate. That is called as the PRPP, right? And what is the enzyme, guys, over here? PRPP synthase. PRPP synthetase, right? So enzyme also you have to mention because I have already told you to mention the enzymes also in the question, right? Biosynthesis, role of key enzymes, and the regulation. That is important, right? So enzyme also you have to mention at each step. From this, yes, guys, again, you already know, yes, we will have the formation of phosphoribosylamine. Phosphoribosylamine, right? You already know the glutamine. It is converted into glutamate, right? Yes, and we have the water involvement. Yes, and the two phosphates will be released, right? 2 PPI will be released, right? Then again, this phosphoribosylamine will be changing into inosine monophosphate, right? And that is the first purine nucleotide which is formed. Very important. First purine nucleotide which is formed, that is the inosine monophosphate. So this is something that they have written from here only, right? So that is you have to remember, guys, right? So yes, all the steps you have to like mention over here. Okay. Hmm. Then again, we have the aspartate and glutamate, right? Yes, the aspartate and glutamate will be acting on the inosine monophosphate. Aspartate and glutamate, right? So we have adenosine monophosphate and glutamine monophosphate, right? Now, yes, so that is something you have to remember. Okay, so yes, these are the inhibitors also, right? The regulation steps. You can like write this points as 6-mercaptopurine inhibits AMP and GMP, right? Yes, AMP synthesis, GMP synthesis, and that is how they are formed. Converted to di and triphosphate, like we have formed the monophosphates. Ultimately, the phosphates will be added to this, and it will be forming the di and the triphosphates, guys, right? So yes, PTP phosphate transfer will be from ATP via NMP kinases, NTP kinases, right? Coming on to the next question, guys. Yes, the 8 marks question. Discuss the role of cholesterol in the human body and explain its biosynthesis. Right? You guys already know this question because we have already discussed in this week pathway of synthesis of cholesterol. All the nucleated cells, especially the liver, the adipose tissue, the adrenal cortex, gonads, the testis and the ovary, and even the intestine. Organelle, it is the smooth endoplasmic reticulum and cytoplasm. Smooth endoplasmic reticulum and cytoplasm. Now, stages of cholesterol biosynthesis. We are asking the role of cholesterol in the human body and explain its biosynthesis. So we are going to explain this biosynthesis also. Yes. Firstly, it is the HMG-CoA, the synthesis of mevalonate, synthesis of isoprenoid unit, squalene, and lastly, how the cholesterol is formed. And we have to like write about all the steps, guys. The HMG-CoA, it is acetyl coenzyme A, acetyl coenzyme A, acetoacetyl. Then again, we are adding acetyl coenzyme A. It will be forming HMG-CoA. Then synthesis of mevalonate from HMG-CoA, which is like from the first step, it will be forming mevalonate. And the enzyme over here, it is HMG-CoA reductase. That is the rate limiting enzyme, guys. That is the rate limiting enzyme. Now, this HMG-CoA can go to cholesterol synthesis. It can also go to ketone body synthesis. It can also go to leucine catabolism. Okay. Now, but for the if he has like this HMG-CoA has to go into the cholesterol, so it is the regulatory step, guys, in the bottleneck of the pathway. After this, if it is converted into malonate, obviously it is going into this cholesterol. Then we have synthesis of isoprenoid units, and then we have the formation of squalene, right? Formation of squalene, that is a 30 carbon compound, and from the squalene, yes, we have lenosterol, zymosterol, desmosterol, and ultimately the cholesterol is formed. That is a 27 carbon compound, and lenosterol, it is the first cyclic compound that is formed. So that is how the biosynthesis is important. And if you can just write about the cholesterol also, the biosynthesis remains the important aspect of the question that we have already covered in detail. Again, all the steps are important, all 5 steps are important with all the enzymes, right? Now, they're asking another question, that is discuss the process of lipid peroxidation and the role of free radicals in cellular damage. Now, you guys already know free radicals, basically, okay, let me just try to write them. Free radical induced peroxidation of membrane lipids in 3 stages: initiation, propagation, and termination. Initiation, propagation, and termination. Now, what is the initiation phase, guys? It involves the removal of hydrogen atom from polyunsaturated fatty acid caused by hydroxyl radical, right? Then we have propagation phase. Under aerobic condition, the fatty acid radical takes up oxygen to form peroxy radical, right? It removes hydrogen atom from another polyunsaturated fatty acid to form the lipid hydroperoxide, right? And guys, you already know the hydroperoxides are capable of further yes, let me just. Yes, these hydroperoxides are capable of further stimulating lipid peroxidation from alkoxy and peroxy radicals, right? So that is you have to remember. And then we have the termination phase, guys, right? Lipid peroxidation proceeds as chain reaction until PUFA gets oxidized, right? So that is something you have to remember, the process of the lipid peroxidation, process of lipid peroxidation, right? Free radicals, guys, you can just remember, yeah, the free radicals, yes. So yes, you already know that during the phagocytosis, the inflammatory cells, particularly macrophages, produce superoxide, right? They convert into H2O2 and hypochlorous acid, and large amount of O2 is consumed by macrophages during their bacterial function, right? So it damages membrane cells and even tissues, right? It damages membrane cells and even tissues, right? So yes, just a sorry for the interruption. Yes. So that is how you have to remember, right? So yes, the preventive antioxidants basically they like the catalase or the glutathione peroxidase, they'll block the initial production of the free radicals, and we have the chain breaking antioxidants also, basically inhibit propagative phase of the lipid peroxidation, right? So that is all that you have to like mention in this the process of lipid peroxidation and the free radical in the cellular damage. Now, guys, the second question, the fifth question that they have asked, that is the functions of the thyroid hormones and the biochemical mechanisms that are involved in their action, right? Let us just discuss about the thyroid hormone functions. You already know, firstly, it is the basal metabolic rate, guys. It is the basal metabolic rate, basal metabolic rate, right? So, metabolism of cells, even in the resting stage, it increases oxygen consumption in all the cells. Again, the metabolism, major catabolic hormone, right? Lipolysis means lipid breakdown, proteolysis, protein breakdown. So it is like it breaks down. It is no, this thyroid is considered hyperglycemic because it is like breaking down the glycogen, and glycogen is broken down to form glucose, and that's why it is called as hyperglycemic also. In the electron transport chain, yes, it acts as an uncoupler as well, right? So person with hypothyroidism have like heat, so it is cold intolerant. Hyperthyroidism have heat, like it's like cold, so it is heat intolerant. Then we have developmental role rate, right? So brain growth, okay, bone growth, congenital hypothyroidism is like more associated with this mental retardation, stunted growth, endocrine system, and it also increases the heart rate and the myocardial activity. So these are something the important aspects, the functions of the thyroid hormones that you can explain, right? Now, guys, coming on to describe the pathway of the cholesterol biosynthesis and its regulation, guys, we have already discussed the pathway of the cholesterol biosynthesis. Now, coming on to just I have discussed the regulation. It is regulated by three ways. You already know we have feedback regulation, feedback inhibition, and hormonal regulation, right? Feedback regulation, it is basically occurring at the genetic level, long-term regulation. You already know, we have already discussed this. If there is enough amount of dietary cholesterol, it decreases the binding of steroid regulatory elementary binding protein at the genes and decrease the expression of genes that synthesize HMG-CoA reductase. That is the rate limiting enzyme you already know, right? Feedback inhibition acting at protein level, right? Increase malonyl weight, it will decrease the HMG-CoA reductase. You already know the yes, the product increases. Obviously, it will have a negative feedback on the substrate, right? And one more important guys, that is the hormonal regulation. You know, insulin is an anabolic hormone. It increases cholesterol synthesis. Well-fed state, insulin, dephosphorylated state. So that is something you have to remember regarding the regulation of the cholesterol. Pathway of cholesterol synthesis we have just discussed, right? A couple of minutes back. Coming on to the next question, they are saying explain the synthesis and degradation of pyrimidine nucleotides. Now, guys, this is like you yeah, the pyrimidine nucleotides, guys, you already know firstly, again, the same thing that we have like discussed over here. Yeah, in purine one, that is like you have to discuss in pyrimidine metabolism also, right? Now, guys, you already know, you just have to mention first about the site, guys. You already know it is the liver. Organelle, you have to mention the organelle. You already know it is cytoplasm and mitochondria. Cytoplasm and mitochondria, right? You already know we have CO2 plus glutamine. That will be forming, yeah, the CPS2 enzyme will be there, the carbomoyl phosphate, right? Then we have the formation of carbomoyl aspartate, right? You already know this. We have an enzyme, guys. We are adding aspartate to this, right? We are adding aspartate to this, and with the help of enzyme aspartate transcarbamylase, right? Now, this carbamyl aspartate will be changing into dihydroorotic acid with the help of enzyme dihydroorotase, right? Now, this dihydroorotic acid will be changing into orotic acid, right? Orotic acid. You already know the enzyme over here, guys. It is dihydroorotate, right? Dihydroorotate dehydrogenase. Then this orotic acid will be changing into orotate monophosphate. And this is this step, guys, from orotic acid to orotate monophosphate with the help of enzyme OPRTase. Yes, it is basically used for UMP like synthase. That is the single polypeptide space. Right? Dehydrogenase, guys, you already know we already have this NAD plus changing into NADH, right? Now this orotate monophosphate will again change into, yes, you already know we have OMP decarboxylase will be changing into uridine monophosphate, then to UDP, okay, then to UTP, then to CTP, signing, yes. So that is something you have to remember the whole process, how this pyrimidine nucleotides are formed. Yes, and guys, you already know the degradation, guys, right? The degradation you already know. We have already discussed the salvage pathway also. We have to just mention that the cell lysis is there, the DNA will be broken down, right? It will be having purine nucleotides. Then what will happen? Phosphate will be removed. Yes, or yeah, the pyrimidine nucleotides are there. Sorry, pyrimidine phosphates will be removed. Then pyrimidine bases will be there. Then obviously the uric acid will be formed. That is something you have to remember about the degradation also. The salt pathways are like mentioned in the notes, and we have like discussed a multiple number of times as well, right? Coming on to the next part, guys, discuss the role of antioxidants in protecting from the oxidative stress. Give examples of key antioxidants. Like you already know, we have the plasma antioxidants, right? We have the cell membrane antioxidants, intracellular antioxidants, and we have the enzymatic antioxidants as well. So yes, these are examples you can like mention over here, right? Now, coming on to the next question, describe the hormonal regulation of blood glucose level, focusing on the role of insulin and glucagon. Now, if you already know, whenever we talk about the insulin, yes, insulin basically it is all on the well-fed state, right? Yes, it is activated in the dephosphorylated state, right? Dephosphorylated state is there. Glucagon, it is the early fasting, phosphorylated state, phosphorylated state. Now, phosphorylated state. Now, this insulin, guys, this insulin, you already know you have to tell this insulin will be increasing glycogenesis, and glucagon is basically will be increasing, yes, glycogenolysis and insulin increasing glycogenesis, right? So these are the pathways that we also have to mention over here, right? Now, coming on to the next one, guys. The functional regulation of lipoproteins that increase, yes, regulation of lipoprotein lipase in the lipid metabolism, and this is something, guys, we already have discussed in kilomicrons, right? We have already discussed this in kilomicrons, and you guys already know that, for example, if even if there is very low density lipids, right? Everywhere, the low density lipids, all the lipoproteins, guys, right? So we have the dietary fat or the exogenous fat, the exogenous fat, right? Now, what will happen? This basically it is in the form of like it will be transported in the blood. We have the triacylglycerol or cholesterol. Yes, we have the three apoproteins which are located, the ApoB48, ApoE, ApoC. If I'm like particularly talking about any chylomicron, and this is something like I've already discussed. This is the structure of a chylomicron. It's a lipoprotein, right? The chylomicron. It will be like, no, these two, ApoB48 and E will identify receptor and attach, right? For example, on the peripheral tissue, we have like adipocytes. So they will come and attach over here, right? And this ApoC is something, which will like activate, yes, LPL, that is the lipoprotein lipase. Very, very important. Lipoprotein lipase. And this is something which will break chylomicron. Or if it is the case of this very low density lipid, just a okay. So yes, this is the lipoprotein lipase which will break this chylomicron, right? And it will like, yeah, very low density lipid. And ultimately, after breaking this chylomicron, we have a very small chylomicron remnant which is left, which will like go into the liver and will metabolize there, right? Little fat is left, right? So that is how you have to explain the function and regulation of this lipoprotein lipase in this case. Coming on to the very short question answers, that is like of 30 marks. Role of carnitine in fatty acid, guys. This is something which you already know and which we have discussed a lot, multiple number of times. That is called as the carnitine shuttle, right? The carnitine shuttle. And guys, how you can just remember it over here. This is all which I have provided. So you already know we have the mitochondria, and we have the cytoplasm of the cell, right? So acyl coenzyme A, which is the activated form of this fatty acid, will be combining with this carnitine in the cytoplasm. Yes. Now what will happen? It will form acyl carnitine, and this coenzyme A will be like chipped off. Now this acyl carnitine can cross the mitochondrial membrane and will come into the mitochondria, acyl carnitine. Now, what will happen? Like what influences the transport? That is the CAT1. Now, this acyl carnitine will be again converting into carnitine. Yes, and it will be combining with this coenzyme A to form acyl coenzyme A, right? And that is by the CAT2 transporter over here. So this is in short about we have to like role of carnitine in fatty acid transportation, activated fatty acid, how it can transport it. So this carnitine is helping acyl coenzyme to come into the mitochondrial matrix for the final synthesis of this, right? So that is the shuttle that they have explained over here, right? Now coming on to the steps involved in the synthesis of deoxyribonucleotides, we have just like discussed that earlier, right? The deoxyribonucleotides. Now coming on to the hormones synthesized by the adrenal cortex. You already know the zona glomerulosa, the three layers, guys. Zona fasciculata and the zona reticularis. Glomerulosa being the outermost layer, guys, and it's a site for the synthesis of mineralocorticoids, chiefly aldosterone. Zona fasciculata, glucocorticoids, cortisol, corticosterone, right? Zonal reticle is production of androgens, right? So that is status you have to mention about all this, and you can just elaborate at all the androgens you can mention, okay, about the aldosterone function, right? The sodium water absorption and all that, right? The salt absorption by the aldosterone, right? So all that we can also mention, even if the names are asked, but just try to mention one line also, so that will enhance your answer. Coming on to the function of the glutathione in cells, guys, you are already aware that glutathione basically is like an extremely important cell protectant, right? It quenches the reactive hydroxyl free radicals. That is one of the important point that you have to remember, right? It's the primary protectant of the skin, lens, and cornea, along with the retina against radiation damage, right? Even the biochemical toxification in the liver and kidneys. GSH basically acts as a cofactor for many enzymes, guys, right? So you have to remember about that also. Apart from that, you can remember it also interferes with the energetics and neurotransmitter synthesis. So these are the basic points that you have to keep in mind while framing the answers. So these are the more important points that like you have to focus on while writing in the answer. Role of HDL in cholesterol, guys. You, I think, already know the role, the reverse cholesterol transport that we have like discussed. Yes, in the last class only, on Saturday only, I have discussed the reverse cholesterol transport. And yes, yes, we have discussed the reverse cholesterol transport. And yes, the reverse cholesterol transport, you already know it is by the HDL only, right? Now, guys, you already know the metabolism of HDL. It is basically from the two sides, guys, right? The intestine and the liver. And we have like discussed this. Yes. So let me just draw. You already know we have the intestine, right? We have the liver part, right? So we have basically all this is coming in the form of a discoidal HDL. It is called as discoidal HDL. Basically, we have cholesterol over here plus phospholipid. Phospholipid, it is arranged in the discoid in the bilayer form. In the bilayer form, right? So that is you already know, and ApoA1 is basically attached. Then we have the LCAT enzyme, right? Now it will convert, okay, all the peripheral organs, right? The peripheral organs, the heart and the other skeletal muscles, they will release cholesterol and cholesterol esters, okay? And obviously this discoidal HDL3 will be now converted into spherical HDL3, which is converting cholesterol and cholesterol esters, right? Now, these cholesterol coming from the peripheral organs come by the transporters. You already know the transporter ABCA1, ABCG1, and SRB1. We have already discussed this, right? ABCA1, yes, ABCG1, then SRB1, right? You already know ABC stands for the ATP binding cassette, okay? And SRB stands for the scavenger receptor B1, right? So obviously this will be converting into like it will be taking yes, it will be going to VLDL and HDL, I mean VLDL, and it will be taking like triacylglycerol in return, and it will be converting into spherical HDL2. That is something called as reverse cholesterol transport, right? And that spherical HDL2 will again go into the liver, these cholesterol esters, and they will be helping in the synthesis of bile, or the bile will be excreted. So that is the reverse cholesterol transport that you have to remember. Inhibitors of cholesterol, right? Yes, the aromatase inhibitors are there, right? You have to remember, right, which compete for binding to the ER and inhibit androgens aromatization, respectively. Obviously, the cyclic part, okay, especially the statins, the rosuvastatin, atorvastatins, they all like block, right, the production of mevalonic acid by inhibiting the enzyme of these three hydroxyl, yes, methyl glutaric coenzyme, enzyme reductase, right? So that is you can remember the metformin hampers the activation of sterol regulatory alimenting binding protein. That is again an inhibitor, right? So that is you can remember, and that how the basic. If you have to treat the yeah, we say now the cholesterol if it's more than 200, 350, something like that. We give statins to lower down the production of the cholesterol, and that statins are basically these rosuvastatins and atorvastatins. Like we have multiple forms, right? term reactive oxygen species, guys, you I think are very well aware of the fact that these are intracellular, yes, these are intracellular chemical species that contain oxygen and yes, and are reactive towards lipids, proteins, and DNA, right? So RF include the suprahydroxide anion, hydrogen peroxide, hydroxyl radicals, right? They are more chemically reactive than oxygen and are able to trigger various biological events. So that is something you have to remember about it. So all the intrinsic properties you can mention, right? So that is how the answer has to be written. Explain the role of ribonucleotide reductase in the nucleotide metabolism, guys. This is like we have already discussed. Primary hormones involved in calcium homeostasis, guys. You have to remember, like, write about the parathyroid hormone. Very, very important. Yes, the physiological aspect, the parathyroid hormone. What does it do to the calcium levels in the blood, in the bone? All that points you have to like mention in bone, in blood. What does it do? Like all those important aspects you have to mention by drawing a flow chart in this particular question, right? And that is how you have to like cover up the question. Structure of a lipoprotein, guys. I think this is the easiest question. You already know it is a compound lipid, right? It is a compound lipid with proteins, with proteins. Now, when I say compound lipid with proteins, so basically this lipoprotein has three layers, right? We have like three layers, right? We have the inner core. Okay, we have the inner core. Basically, it is consisting of hydrophobic tails, hydrophobic lipids, right? And these hydrophobic lipids are these triacyl glycerol. Obviously, if they are hydrophobic, yes, these are triacyl glycerol. Or I can say the cholesterol esters. Cholesterol esters. Then we have the second layer, which is now consisting of, yes, empathic lipids. Empathic lipids. And that is basically consisting of this cholesterol and phospholipids. Phospholipids. And third layer, basically, it is of proteins. It is of proteins. Okay, we have peripheral proteins. We have peripheral proteins. And we have integral proteins. Integral proteins. And that is like ApoB100, like that. Okay. So that is from outer to inner. I mean, from the inner core to the outer one, you can see we have the inner core that is like the triacyl cholesterol esters, right? The triacyl glycerol and the cholesterol esters. Then we have the second layer that is made up of empathic lipids. Then we have the proteins which are made up of like peripheral proteins are there, right? And integral proteins are there. So that is the basic structure from outer to inner, and that is like you can draw a diagram and you can just explain the question in this particular manner as well. Right now, coming on to the next part of the paper, that is the multiple choice question, which is the following is an essential fatty acid. That is something you already know. Yes, the linoleic acid is the essential one, right? HMG-CoA reductase multiple times now. It has been, you already know the cholesterol synthesis, right? In the synthesis of the purine nucleotides, it is vitamin C, guys. Primary antioxidant, yes, it is vitamin. Hmm. Yes, it's, yeah. Hmm. Again, okay. Let me just discuss this first. Yes, vitamins acts as the coenzyme in the synthesis of purine nucleotides. Yes, it's the vitamin C. Yes. And yes, so it is like vitamin K, right? The urea cycle primarily occurs in which organ? Yes, you already know it is the liver, again from the protein catabolism, right? So we have again drawn the liver in the like the earlier mock test which I discussed on Saturday. It's like that. Which hormone increases the activity of the hormone sensitive lipase? I've told you it is the insulin which increases the activity of the hormone sensitive lipase, right? The end product of purine catabolism in humans, it is the uric acid. The key regulatory enzyme in glycolysis, it is phosphofructokinase. That is the rate limiting enzyme. Yes. Which of the following is a marker of the oxidation state? Guys, remember it is melon-D-aldehyde, right? It's the key marker of this oxidative stress. Others are not. And the last one, which of the following hormone is derived from cholesterol? Guys, yes, you already know we have all the steroid hormones. They are basically derived from the cholesterol, guys, right? So that is regarding this paper that we have like already sorted off, because we have already discussed this a lot number of times. All the questions are being discussed in the class. Like I've explained you in detail, guys, all the questions that we have like studied over here, right? So yes, that was about today's lecture, in which we have like focused on all the important questions that have been asked in the revised schedule part, like in the previous sessions, right, in the previous examinations, and like multiple times I have asked in many universities, guys, right? So like you have to focus on how to write the exam, guys. How much to write? Actually, I have already told you. Whatever the question is asking, please, in how many parts, just try to put the heading and complete that part first. Then the next part of the question, put the heading and then complete it. Better to draw a flow chart wherever needed. All the steps, please write it down in one single flow chart. All the enzymes with other different pen, and try to like mark all the necessary coenzymes that are required over there, right? So that is regarding today's exam. Hmm. So yes, we have already focused on all the questions above. We have like discussed. Yeah. So all the MCQs like are very easy. You can just go through that. Yes. And yes, these are the social media handles, guys. You can just follow for the further updates of the classes, right? And for further live classes. Thank you. Thank you, guys.